Hi everyone, it's a real pleasure to have Alipasha Vaziri here as a speaker. So I wanted to just tell you a bit about his trajectory. And that starts with a master's and a PhD in quantum physics on quantum entanglement. And he told me a story a long time ago that I remembered, how he discovered the first experimental confirmation of quantum entanglement of orbital angular momentum of a high energy photon that decays into two lower energy photons. This was late at night in Vienna, and he had to walk back to his house because the public transport was already down. So he had this profound hour to ponder over his discovery of this thing that at that point in time only he had ever known about in the world, until he told his advisor the next day. I thought that was a spectacular start of his scientific career, and since then he's been going and doing ever more interesting things in biology and also physics and microscopy. But he did take a break of 2 years and worked for McKinsey on strategic business consulting, more or less to see how that world worked, then quickly came back to science, did a postdoc with Chuck Shank at Geneva in the early days of Geneva. And here started working with biologists like Jeff McGee, of course, Chuck Shank and Carl Svoboda, and started using two-photon techniques, temporal focusing, to do multi-layer super resolution microscopy, and also a single cell optogenetics. And then got a position at IMP Vienna and a professorship at the University of Vienna, and kept working on optics and biology using temporal focusing for larger scale recording and optogenetics, and then also developed light field microscopy for use in C. elegans and larval zebrafish to start doing larger scale imaging, whole brain imaging at high rates. Then moved to Rockefeller University, where he continued his work on microscopy and work on biology in his lab and in collaboration with other labs, and moved to more complex biological systems, from cell culture and slices to in vivo C. elegans and zebrafish, and now in mouse and marmosets, to harder to access scattering tissues, and really takes challenges that are hard and solves them. I think we'll hear about some really challenging microscopy techniques that his lab just got to work in a great, very interesting and very ambitious way, that now allows for the very large-scale imaging of neural activity in a variety of animals. With that, I really look forward to your talk. Thanks again for coming.
Thank you so much, Misha. Yeah, thank you again, Misha, for this very nice introduction. It's great to be back here after such a long time, and also it's great to be able to give finally a person seminar after 25 months of COVID. So I'm very much looking forward to meeting with more of you and interacting throughout the next day or two. So as you heard from Misha, the focus of my lab is the development and application of neurotechnologies that have been aimed at pushing the scales at which neural populations can be recorded in terms of speed, resolution, or scale. And over the years, we have tried to push these technologies from smaller model systems to larger ones. And today I would want to give you an overview of some of these technologies, and perhaps a little bit of an idea on what kind of qualitatively new types of neurobiological problems they allow us to access. So what you're seeing here is a recording from a single plane in the mouse visual cortex containing about 70 neurons. It is done by a conventional two-photon microscope that is commercially available, and these days has become very standard tool in many neuroscience labs. In comparison, this is the scale of our most recent developed technology that we call light beads microscopy, that is now covering about 1 million neurons distributed across both hemispheres of a mouse brain, and covering a volume of 6 by 5.4 by 0.5 millimeter. So as you can see here, the relative scale of these two volumes or neuronal populations is basically what I want to give you an idea about on what it takes to scale up imaging to that number of neurons. But really, from a biological perspective, the motivation of a lot of our work is founded in some of the questions that are rooted ultimately in the neurobiology that is in no means unique to our lab, but they are the following. So we are interested in understanding how sensory inputs are represented across the brain. How does the distributed and large-scale activity of the neural population generates behavior? And what are the computational principles ultimately that underlie generation of behavior and the various brain functions? So to address these questions, we can maybe focus on the first one, and I can ask even a simpler question, and that is, how is the information distributed within its encoding neural population? So in other words, what I can ask is, if I look at the amount of encoded information in a neuron population as a function of neuron number, what is the scaling here? As I keep adding more and more neuron, do I encode more information, or what's the scaling law here? So one possibility, as you can imagine, is that we could be facing an unbounded scaling. So we simply encode more information as we add more neurons. However, there is also evidence and the possibility that as we keep adding more neurons, we may be saturating ultimately the amount of information that a population encodes. So we could be facing a bounded scaling. The problem, however, with the commercial tools that I mentioned that are available is that we are basically in this regime. So there is no way for us now to know in which of these scaling laws we are moving forward. But really knowing that information, for instance, knowing how sensory inputs are represented initially among the primary sensory areas is key, because that will also determine on which type of information some of the downstream areas have access to. So this has not been possible with a small number of neurons recordings. Another reason is that also that there is more evidence for that, is that there are different states of an animal that are differentially represented. For instance, we know that movement-related activity is very redundantly and widespread across the brain, whereas perhaps some of these more cognitive states related to decisions or so can be represented by very small number of neurons that could be very sparsely distributed. So really having these large-scale tools allows really to survey the brain and discovers maybe some of those more hidden signals. So this is one motivation. The other that also our lab is very much interested in is related to the variability of neuronal responses across different trials as we look at the animal in behavior. So more concretely, for instance, imagine these would be 3 sensory tuned neurons that are now, for the sake of the argument, would be in brain regions that are sufficiently far from each other that we cannot record them simultaneously. So what that means is that we can only look at the average activities of these tuned neurons, and based on that, we may be believed to assume that they are doing the same thing. However, if we were able to record them all at the same time, we would see that there is tremendous trial-to-trial variability of these responses for the same sensory inputs. And so this has been documented extensively. And these are basically what is referred to as noise correlations. So they are thought to be representing internal states of the animal that are together with the sensory inputs, and the motor states are responsible for changes also in the behavior. So these kinds of tools allow us really to capture these noise correlations and allow us to look at how, for instance, decisions unfold on a trial-by-trial level in animals. So along these lines, we have been developing initially tools that had allowed us to manipulate neuroactivity in a targeted manner, but really in the last 10 years or so, our main focus has been on the development of parallel and unbiased recording technologies for large neural populations. And as Misha was pointing out, we have been trying to push those from smaller and more transparent organisms, such as the C. elegans and the larva zebrafish, and more recently into the more scattering brains. Now, I don't think to tell this audience that really the key enabling factor for why we can even develop these optical neurotechnologies has been the tremendous advances on the molecular side and the genetic tools, in part what has been achieved at Geneva with the development of GCAMP that has really enabled us. So our work is mainly addressing the challenges on the optics side. So, and not only in our lab, but there are a number of other different technologies that have been developed by other groups. So, and one way maybe to categorize them is to look at them along the dimension on how sequential versus parallel their excitations are, right? So you can imagine a fully sequential approach, like a point scanning that requires you to scan in 3 dimensions to generate information about the volume, like confocal or 2-photon microscopy. And there is a spectrum for other methods, such as, for instance, projective approaches, exciting along one line or a sheet, etc. So in all of these approaches, what we can conclude is that the volume acquisition time is proportional to the exponent of the pixel dwell time and the degree of the sequentiality of this approach. So as we go to more and more sequential approaches, and for larger and larger volume, eventually these approaches become unscalable because of this inverse relationship between the speed and the volume sizes that can be accessed. Now we could ask, okay, what if we take it to the extreme? What if we excite the entire volume all at once? So this can be done, but then we have to solve a different problem, and that is, how can information from each of these volume pixels be recorded and assigned in 3D to the individual sample positions? So this is one approach where this coupling of the acquisition time and the volume size can be solved. And one solution to that we have looked at a while ago is this idea of light field microscopy. So the best way to illustrate that is, maybe you have seen one of these cameras. So they are having an array of lenses, and the very fact that each lens is positioned at slightly different positions in space means that when you capture a photo, they all capture different angular views from your scene. So you end up with these different angular views of your object, and then you can computationally infer the volume information. So in this case, the number of perspectives is related to the number of lenses, also related to the axial resolution that you can get, and the lateral resolution is related to the number of pixels behind each lens. So the advantages of this approach is that it is intrinsically instantaneous. So with a single snapshot, you map the entire 3D volume positions into patterns in 2D. So there is no mechanical and scanning of moving components required, and it is, at least on the hardware side, relatively simple to implement. I just want to also point out that the origins of that actually go back, are quite old, to 1908, to Lippmann, so preceding even confocal microscopy, but in the more recent era, it has been popularized by Edelson and taken up by Marc Levoy, mainly in the context of photography, but more recent groups have adapted this also in the context of microscopy. So a few years ago, we decided to build a microscope based on this idea. And this was the work of a former postdoc in the lab, Robert Prevedel, and a student, Maximilian Hoffmann. And we teamed up with the lab of Ed Boyden on the computational side and developed this tool. So the basic idea here is that we can design a micron lens array that we can put in the image plane of a conventional microscope. And what it does is that it leads to an encoding of sample positions in 3D onto patterns of excitation on the array. For instance, if the point is in focus, we get excitations behind a given microlens, but where the point is above or below the focal point, this leads to excitation of patterns that are very densely coding for the position within the sample. So what we can do is we have now this information about the optical system and the encoding, how we go from the volume to these light field images. And during the reconstruction, we are basically trying to solve the inverse problem. We are asking, given these images, where are the most likely locations of their emitters? And one way this can be done is by doing deconvolution and solving this inverse problem that can be formulated in the context of a Richardson-Lucy algorithm for deconvolution. So we did it, and we could get information about individual neural activities that works relatively well in semi-transparent systems such as fish and the worm. However, due to this intrinsic fact that we are trading off angular and spatial information, you will never get to the diffraction limited information, and you're basically trading off these two parameters against each other when you are doing light field imaging. However, what we realized is that in case of neuroactivity, we are not really imaging static objects. So these are not objects that are activated or on all the time. So the fact that some neurons are sometimes on, sometimes off, we thought might provide us with an additional degree of ability to better localize neurons. And so we came up with this new approach that we call seeded iterative demixing, or SID. So the basic algorithm here is such that we get this light field images, we perform a background subtraction, and then calculate a standard deviation image, which is basically capturing the most of the varying information in the sample that is correlated with the neuroactivity. We then deconvolve, do a reconstruction using deconvolution in the same way as I was mentioning before, to get into the 3D space and perform a segmentation of these 3D locations within the volume, and treat these segmented regions basically as putative locations where neurons could be. We then project them back to the camera sensor, getting these what we call spatial footprints of neurons, basically how a given cell body could look like on the camera, and then compare it to the raw data that we record with the camera to get a first estimate of the time traces, and then we compare the time traces to the raw data again to get an update of our initial guesses of the footprints. So together we are solving a constrained biconvex least square optimization problem where the individual temporal and the spatial components are known to converge. So we implemented that and then looked at, for instance, the larval zebrafish brain. So that allows us now to look at the volume that is capturing the majority of the brain of the animal on the order of 700 by 700 by 200 microns. And in this case, we can, in principle, go as fast as our camera rate allows us, and as long as we have enough photons. So we typically image at rates on the order of 10 to 20 hertz when we are using GCamp. So this is sort of the type of information that you get after running SID on these neuronal time traces. So you can see, in most cases, relatively well localized and information to individual neurons that you can see in both in 3 dimensions. So we developed that a while ago, and then a few years later, a postdoc in the lab, Xian Lin, was interested in applying this tool to what I was mentioning in the beginning of talk, sort of how decision-making in larval fish evolves on the trial-by-trial level. Since we can now capture the entire brain relatively fast, we thought that could be a good application of this tool. To study decision-making, however, you need a paradigm for the animal to learn and to be able to perform decisions, and that has been difficult to establish. So we adapted this Operant learning assay that was originally developed by the lab of Florian Engert that is referred to as ROAST. So the basic idea is that these larval fish are embedded in agros, their tail can move, and they are receiving a slightly aversive stimulus in form of a heat response delivered by a laser. And they are released from the heat stimulus if they make a correct tail movement in the target direction. So the animals can learn this behavior after a few trials, and then you can also reverse the order of the training and do, in this case, a left training, and they would relearn the trial. So we performed this type of training and looked at the learning progression of the animal and how the decision evolved on the single trial level while recording from the whole brain. So here you see the time traces, now the heat maps of these about 5,000 neurons overlaid with the tail position of the animal, and as well as the delivery of the heat stimulus. And when we looked then now at the neuronal trajectories and how they evolved in the phase space, we found that on the individual trial level, they started from a common location and evolved together that was encoding basically the heat representation. But well before the action selection, there was a bifurcation of these trajectories on the level of individual trials towards two different outcomes that were predictive of whether the animal would be moving to the right or to the left. So to better understand this, we performed dimensionality reduction on these time traces, and it did clustering and mapped the clusters back to the anatomical locations of the animal's brain, which are shown here, as you can see. Now, since each of these neurons could have, in principle, a mixed response, sometimes to different sensory inputs, sometimes to behavioral outputs, to decouple those contributions, we performed the demixed DPCA, or demixed principal component analysis, that allowed us really to investigate the contribution of each neuron to decision-dependent or decision-independent parameters, and then investigated the response of each of these clusters along these two dimensions. So one of the surprising findings here in this study was that the cluster 4, 2, and 5 together, which are the bihemispheric cerebellum and ARTR neurons, seem to be encoding for both aspects of the decision. So here you can see the first DPC component of the decision-independent variable. And what we found is that the ramping rate from the heat onset before the motion execution was predictive of the time point when the animal would be doing the decision. And similarly, on the decision-dependent components, the bifurcation of these trajectories, as I was showing you before, after the heat onset, was predictive of the direction of the animal. And this could be done about 10 seconds in advance before the animal was executing the behavior. So I show you that a little bit on an individual trial. What you see here is the activity of the neurons. Here you see when the heat comes on, and our moment-to-moment prediction of the turn direction, the tail position, and then our prediction of the turn time as reported by the prediction error. So at some point the heat comes on here. Yeah, so we predict right, left, right again. The prediction error drops. So at this point, we are predicting the animal would be doing a right tail movement at time point 0. And if we wait, yeah, we see the animal is actually doing that tail movement. So this was one example to show that that this tool allows us to look at the outcomes on the individual trial level. But we asked, is this really necessary? What if we wouldn't have had the ability to record from the whole brain? So to do so, we compared now the same predictions or the same questions with the fact if we would use a 2-photon single plane recording. In this case, we assume that one would have access to about 180 neurons. So this is the separation of the trajectories, as I was showing you before, for the turn direction. But if we reduce that to 180 neurons, the trajectories are simply not separated enough from each other that would allow us to do a reliable prediction, because the amount of information is highly distributed in the population of these neurons in the cerebellum. So we couldn't do a prediction of the turn direction with 180 neurons. Similarly, if we look at the prediction of the turn direction, this is what I showed you before, and with 180 neurons, at 10 seconds before the movement initiation, the prediction error is still very high. So we cannot do a reliable prediction also of the time point of the turn.
Sorry, excuse me. Is this 180 randomly chosen neurons, or what happens if you choose the 180 most informative neurons?
These would be, so we pretend. I think the question was, were they 180 targeted neurons or 180 randomly selected neurons? Is that?
And I was wondering, if you do choose the 180 most informative neurons, how well can you predict it? Because you were saying that it was very distributed.
Yeah. So I think in this case, we recorded even from 180 neurons in the in this cluster, which we a priori knew that they would be contributing to that. So the cluster 4 and 5 that I was showing you before, not throughout the entire brain. So even doing that in a single plane gave us this result. Now, if you would, for instance, now go in and rank the neurons depending on their contribution, and then go in and then look from those 180 neurons, that's not something that we have done. But in order to do that, you would need access to this other tool of being able to record all of them. So we wanted to make the case, what if you wouldn't have this tool? What if you just do a single plane recording with a conventional microscope? What would be lost? This is just one example, and I didn't go so much into the biology, but we have a few follow-up questions now, and trying to understand what is the origin of this ramping activity in the cerebellum. We think it has maybe something to do with interaction with habenula, and there are some follow-up projects on that the lab is looking at currently. Okay, going back again to the technology. So after recording throughout the brain in smaller systems such as C. elegans and the larval zebrafish, we shifted our focus and asked, what about extension of some of these technologies to the more scattering mammalian brain? And I think you all know that the standard tool has become for imaging mouse brain is 2-photon scanning microscopy. But there are some fundamental limitations in terms of the scalability that I also mentioned at the beginning of the talk, which I want to walk you through briefly. So let's imagine that we want to image the activity of neurons within a volume that is only 500 micron cubed. And this would be the size of our diffraction limited spot. And let's say we want to do that at the rate of 5 hertz. So simple math shows us that translates to 500 million voxels. Sampled at 5 hertz means that we need to have a laser that runs at least at 2.5 gigahertz in order to excite each pixel or voxel at least with one laser pulse. So such a laser is not conventionally available, but even if it would be, there is a bigger problem, and that's because the dwell time now at this acquisition becomes an order of magnitude shorter than the fluorescence lifetime. So we wouldn't be able to collect any photons from these excitations. So we can ask, can we relax the spatial resolution without losing information? And so if we look, at least if our goal is to record at cellular resolution in neurons in the brain and look at the size of the cellular size of basically stomata in the mouse cortex, they are on the order of 10 to 15 microns. So in some sense, you may think that the 0.5 lateral size of excitation is maybe unnecessarily high. And in fact, what people are interested in is the axial sectioning capability. So maybe we can relax the spatial, the lateral size of the spatial excitation to something that would be about 5 micron, in which case we would be still sampling objects that are 2 or 3 times larger faithfully in terms of Nyquist criterion. The problem with that, however, is that the beam will be extending over 70 microns axially, so we would lose all optical sectioning in this case. So from this, you can see that what would be beneficial is to be able to have a more comparable lateral to axial excitation that is overall maybe a third to a half of the size of the objects that we are interested in. But in order to achieve that, you basically need to be able to decouple the lateral and the axial confinement of excitation. And that brings us to this idea of light sculpting, or shaping the distribution of light in 3D in a more arbitrary way. So that is exactly the idea. So if you think of a conventional Gaussian beam, the 2 key parameters that are defining it is the spot size d and its axial confinement depth of field or relay length z. And both of them are determined by the wavelength of light and the numerical aperture of the optics. So as such, there is an inherent coupling between them. So whenever you focus the beam to a certain size, you end up with a certain axial extent. And what we are asking is, how can we go beyond that and create something more arbitrary? Now, one way that this can be done that has been used by our lab and also other labs in the recent years is to use temporal focusing as the means to do light sculpting. So the basic idea here is that if you are using these femtosecond optical pulses, there is another degree of freedom that comes with them, in a sense, that has not been used so much, which is the bandwidth of the spectrum that they contain. So the basic idea in temporal focusing is that you can send these pulses to a dispersive element, such as a grating, which sends then the different spectral components or colors along different spatial directions. So as a given pulse is propagating away from the grating, the peak intensity drops off as a function of propagation. And then you can reverse the process by setting up a telescope that is configured such that is imaging the spot on the grating where the pulse was initially short back again to a given axial location in the sample, right? So with this arrangement, you bring together again all the spectral components of the pulse, both in time and space, to a given axial position, and recreate this peak intensity of the pulse that can effectively excite the fluorophores. But above and below that target plane, you're having a geometrically chirped pulse that is not effective in exciting the fluorophores. So what you have achieved, in a sense, is you can use the spectral bandwidth to control the axial localization of the beam, while the lateral localization of the beam can still be controlled by the illumination spot, the size of the illumination on the grating, and the demagnification ratio of the telescope that you have. So just as an overview, in conventional two-photon, you create both localization laterally and axially just by focusing in space. And in this case, you vary the pulse width as a function of axial direction or time, and thereby can control the axial confinement to a different degree of freedom. So this is just a simple example that we've shown on a fluorescent slide. So you can create, in this case, a beam that's maybe 10 microns or so wide, but can axially still be confined to something as little as 2 or 3 microns. But as I said, the degrees of freedom are relatively decoupled. So we set up this idea, going back to the question that I was posing before. And this is the work of another postdoc in the lab, Siegfried Weisenburger, who set up this scanned temporal focusing microscope. So we had a excitation spot on the order of 5 micron, an axial confinement of about 15 micron, and then the beam was scanned laterally and axially to cover the volume. And now if I go back to the same example that I was showing you before, you see that the whole volume translates now to 1 million voxels sampled at 5 hertz and 1 pulse per pixel, which ends up being the most efficient way how you can create fluorescence per input average power. It means that you need a laser that runs only at 5 megahertz repetition rate, which is now more conventionally available. And more importantly, the voxel dwell time is now 200 nanoseconds, so you have enough time to get the fluorescence light out. So we created this setup of the scan temporal focusing, and then added a few other sort of additions that helped further speeding up the acquisition in a volumetric sense. So one was this idea of remote focusing that was originally proposed and shown by Tony Wilson. So the idea here is, instead of moving the objective up and down, which can be slow and also introduce vibrations, we can create a conjugate plane to the sample where we can place a light-weighted mirror, in this case that can be positioned on a voice coil, and that can be then moved very rapidly up and down. Thereby, we will be changing the divergence of the beam into the objective and scanning that plane rapidly up and down. And the third one is this idea of temporal multiplexing that has been also again used originally almost 10 years ago or longer, and then more recently have been also adapted by a number of other groups. So I want to briefly outline the idea here. So if you look now at the separation of our pulses of this 5 megahertz repetition laser, they are separated by about 200 nanoseconds, right? And when they excite the sample, the fluorescence is emitted after 6 or 8 nanoseconds after the pulse hits the sample. So if you look at that, for the majority of the time, there is no exchange of photons between the sample and the microscope. So we are just waiting for the next pulse to arrive, which is not very efficient. So we can ask, wouldn't it be much better if we had just the next pulse coming right after the fluorescence has decayed, and then the next pulse, and so on? So that is the key idea. The way that can be created is that we can start off with a much stronger pulse, split it by sending it to a series of beam splitters, and delay each individual beam by a multiple of the fluorescence lifetime, and then bring the beams again together, and then we can direct them towards different axial positions or different lateral positions, all of which then can be sampled by the same mirrors. And then all we need to do is to look at the arrival times of the photons. And by knowing when the photons have been arriving on the PMT, we will be able to assign them to the individual planes. So we can speed up, in this case, acquisition by fourfold. So you put all of these ideas together, and then in this case, looked at mice that could run on a treadmill, while we looked at the brain through a window. We could track the position of the treadmill, as well as we could provide them with different somatosensory stimulation, visual stimulations, or auditory stimulations. So to demonstrate the functionality of the axial multiplexing, when the sub-volumes were on top of each other, we recorded from the PPC, targeting a volume that is about 500 by 500 by 600 microns. And for the lateral multiplexing, we looked at the primary auditory cortex. So this is now the representation of the activity when we have the axial multiplexing. You see the sub-volumes. It spans a volume of about 700 by 700 by 600 microns. And we can record now 6,000 neurons in this configuration at 17 hertz. We can also reposition these volumes such that they are next to each other. In this case, they are spanning a volume of 1 millimeter by 1 millimeter by 600 microns, containing 12,000 neurons in the auditory cortex that we can sample at 5 hertz. And so this is how the heat map looks like. And in these cases, they start becoming not so informative anymore because of the density of the information. But if you zoom in, let's say in 200 of these, you can see that we can get relatively good signal to noise ratio for the individual traces and of calcium response times that are comparable with other techniques that people are familiar with. So in this case, we provided the animal with different auditory inputs in the range of 6 to 81 kilohertz. And then we could find different auditory bands to which the animals, groups of neurons were responsive to. And on the individual neuronal level, we could see that individual neurons were having a preferred auditory response of auditory frequency, but they also somewhat responded to some of the neighboring frequencies. And so this is now being pursued by another graduate student in the lab, who is now with this tool interested in understanding what is the interaction now with different multiple auditory inputs. So, and we find interesting observations about how they are non-linearly now being integrated if I provide, for instance, 6 and 20 kilohertz at the same time, and so on. So we are trying to build up more complex auditory percepts and stimuli bottom up, and as well as looking in a top-down manner. In terms of the imaging depth, all of what I've been showing you so far has been reaching to maybe something on the order of 600 microns. But you will note that the brain is scattering. So as we try to image deeper and deeper, the number of ballistic photons that are reaching the focus starts to decay. And so they will ultimately go to a level that the signal to background ratio reaches 1, because as we are trying to compensate for this loss, we start at some point exciting fluorophores on the surface. So, and that happens in the mouse cortex typically in the range of maybe 800 microns or so. So at that point, a further increase in the power doesn't help. So we need just to switch to a higher nonlinearity and do 3-photon excitation. And so we built a 3-photon excitation arm to this multiplex scanned temporal focusing tool that was running at 1300 nanometer. So it was synchronized because we had the same pump laser that was seeding another OPCPA to create the 1300 nanometer light. And we overlaid the beam with the 2-photon beam using a dichroic, and did similar type of optimizations in terms of point spread function engineering, but we didn't implement temporal focusing in this case. So with that 3-photon excitation, we can go now throughout all the layers of the cortex through the corpus callosum and image volumetrically the CA1 neurons of the hippocampus. So here you see some example planes at 1.16 millimeter or 1.22 millimeter of the CA1 and the corresponding neuronal time traces. Now, because of this 1 pulse per pixel excitation scheme that I was mentioning to you, we had an acquisition rate of a single plane of 78 hertz, which you can see here that still under these conditions, while the images are not very sharp, you can still identify cell bodies relatively well, and the signal to noise and the signal to background ratio is acceptable. So we use the extra overhead that we had in terms of time here to instead look at the volume. And this is now the first volumetric imaging that is done using 3-photon microscopy of about 200 neurons recorded at 4 hertz. And we are looking here at the depth range of 750 to 1000 micrometer in the mouse brain. And finally, you can imagine that you want to put your resources in the best, most efficient way. So to do 2-photon microscopy for neurons that are on the superficial layers of the brain, and then 3-photon for the deeper layers. So we came up with this hybrid configuration where we could apply the multiplex scan temporal focusing to look at the activities of neuron across the different layers of the cortex, and at the same time look at the volumetric information of the neurons in the underlying CA1 neurons of the hippocampus. So in this case, we are capturing 3,500 neurons at 14 and 5 hertz respectively, reaching an axial range of a total of 1100 micrometer roughly. So one of the interesting applications of this tool is, for instance, that it allows you now to look at the correlational structures of neurons across different axial regions. So we can look at the neuronal correlations across depth. And what we found is that there are a significant amount of correlation within each layer of the cortex, as well as across different layers of the cortex, but also between the hippocampus and the cortex. And this can have applications, for instance, in validating some of the models that people have been proposing before about canonical circuits of computation within across the cortical layers. And that is sort of one application for which we have developed this. And there are a few projects that we are collaborating now with a few biologists. Just want to also point out that as of last year, we have licensed this tool. So we hope that at some point there will be also a commercial version of that people can use. Okay, so stepping a little bit back, we started to see, okay, what is it that we have learned in terms of design concepts for scalable volumetric calcium imaging? I mentioned some of them throughout the talk, but so one of them is that each sample voxel should be excited by exactly one laser pulse. The reason for that is that you can gain non-linearly in time in terms of the signal for the same amount of average power that you're putting into your sample. Then the choice of the lateral and the axial PSF for a fixed power budget is determined by the trade-off between the sensitivity and the neuronal detection versus the total number of detected neurons or the size of the volume that you're interested in looking at. So depending on whether it's more important for you to record as many neurons as possible or to detect every single neuron within the volume that you're interested in recording, there is a trade-off here. The next one is that the spatial sampling should be also done at Nyquist limit with a voxel spacing that should be comparable to the lateral size of the excitation roughly. And the excitation of adjacent voxels should be separated only by the fluorescence lifetime, but not more. So if you look at these criteria, most of them, basically the first 3 have been fulfilled by the methods that I was showing you before. But the last one wasn't all the way fulfilled. And the reason for that is that if I show you the time scales that I was showing you, this axis was actually broken. So if I show you now in scale, you see that 168 nanosecond out of the 200 nanosecond, or 85% of the interpulse interval had remained still unexploited. So you can ask, why can't we repeat that? Why can't we create yet another delay and so on? And the reason is that if you run the math and look at how many pulses would it take, it would take about 32 or 30 pulses to span the entire interpulse interval. And the total length, associated length with the delay line that you need is almost a kilometer. So that is not very practical for setting up in a lab. So we came up with a different approach and idea. And this is the work of another postdoc, Jeff Demas. So we came up with this cavity-based design. So the basic idea here is that you start off with a higher energetic pulse from a laser, and you couple it on top of an output coupler into an 8F re-imaging cavity. So the cavity has a focus here and another focus here, and then it has an intentional axial offset that is shown here as DZ. So as the beam is coming back towards the output, the beam starts to diverge, right? And also it's tilted back downwards a little bit such that it hits the output coupler. A portion of it is coupled out, and the rest of the beam undergoes another round trip, and so on. So what this cavity does uniquely are achieving 3 goals all at the same time. So first, the total length of the propagation of the beam here in the cavity has been chosen such to correspond to the desired delay that you want to create in time between the pulses, so on the order of 7 or 8 nanoseconds. The second thing is that through this axial offset, through each round trip, each of the beams accumulates an amount of divergence, as is shown here. So as their beams are being coupled out and are focused by a lens into the sample, the subsequent beams start focusing at shallower and shallower depths. And the third is that the reflectivity to transmission ratio of this output coupler can be chosen exactly such to compensate for the loss of ballistic photons in the focus. So what you end up with here is basically a series of 30 axially focused spots that we refer to as light beads. And each of them arrive at a delay of 8 nanoseconds or 7 nanoseconds with respect to each other. And the pulse energy in all of them starts to increase in an exponential way, such that it exactly compensates for the loss of photons due to scattering. So you have the same signal to noise ratio in all of them. So all you need to do is basically you need then just to scan in 2D and collect the photons. And we coupled that to a mesoscope because we wanted to take advantage of the speed along the axial resolution to capture as large of a volume as possible. And this is the movie that I was showing at the beginning of the talk, through which we can now, having access to a field of view of about 6 millimeter, we can record these bihemispheric activities of neurons at different depths in the mouse brain. So if you look at the individual time traces now, this is just a representation of these neurons. They are shown here again. So this is now for another recording that is 800,000 neurons. But again, these heat maps are very uninformative, and we can start zooming in here. But again, as you zoom in, it's little information that can be gleaned from these representations until you get more to the cellular level. And here, as you zoom in onto the level of individual neurons, you start seeing that they do have representative responses of activity. So this is very early days, and we are still exploring the data. It's very large and what we can learn and apply to different questions, but I would just want to give you a couple of ideas. So one thing you can do is you can look, for instance, at correlation structures within this large population across the brain. So this is now, if I now look at a large portion of these neurons on the order of, say, 36,000 of them, if I look at the distances of the correlation, exhibit correlations that are more than 2 millimeters, supporting this idea that information has a very widespread multi-regional representation across the brain. We also use now at how are now, for instance, individual sensory modalities represented with this tool. So we looked at the smaller volume and specific brain regions. In this case, we looked at a volume that is 3 by 5 by 0.5 millimeter in size, and which spanned the visual cortex, somatosensory cortices, the retrosplenial, etc. And in this case, we provided the animal with repeated visual stimuli, somatosensory stimuli, and combined visual and somatosensory stimuli, while the animal could run freely on a treadmill. So what, unsurprisingly, we found neurons then that were tuned to viscous stimulation, some that were tuned to the visual, and then some that were tuned to the behavioral state of the animal, and then some that were uncorrelated. So these are just some example neurons. We could then hierarchically cluster the correlation matrix of these neurons, sort them across 4 different clusters, and sort them across the preferred stimuli, behavior or stimulus, viscous tuned, visual, or behaviorally tuned neurons, which you see here. And then if we now map these correlations onto the anatomical locations of the brain, we found that each of the 4 clusters were represented in each of these modalities. But for instance, for the viscotuned neurons, we found them overlapping more with the somatosensory regions, for the visual, more in the visual cortex. One observation was that the behaviorally tuned neurons were really widespread throughout the entire brain and were very large in their number, and also represented across multiple clusters. And so were also the uncorrelated neurons. And we saw evidence for a lot of mixed selectivity. So each neuron didn't have necessarily only a unique response. They could respond to different aspects of the stimulation. And moreover, they were also cross-modulated. So in this case, for instance, if I look at viscotuned neurons, we saw their responses to be modulated by the presentation of the visual inputs, with some groups of them that were spatially segregated, with a group showing an upregulation of the response and another group being negatively modulated. The same for the visually tuned neurons were modulated by viscous stimulation, and both modalities were also modulated by the behavioral state. Now, again, so this shows this kind of tuning maps and their mixed selectivity, but you can argue, okay, if we didn't have this tool, couldn't we record this information in a sequential manner? And you may be able to get tuning maps also in a sequential manner by doing multi-plane recordings. But one element that would be lost, again, is this trial-by-trial variability of the responses to the same sensory stimuli. If I look, for instance, now here at viscotuned neurons, a group of viscotuned neurons modulated by the visual stimuli, I can find now groups of neurons whose responses are co-varying with each other. So these 4 neurons' responses are co-varying with each other, and they are separated by more than 1 millimeter. So I wouldn't be able to capture that otherwise. But there are also a group of them, these other 4, that are not exhibiting any co-variation. So these are additional informations that we are now able to record from the brain, and we are interested in exploring further. Okay, so again, just want to step back a little bit and say what is it that we have done. So I have shown you that we have been able to push the volumetric recordings over the last 10 years or so from small model systems. And then in the more recent years, the number of neurons that we can record keep increasing. So if I were to naively extrapolate for this and being very optimistic, I can say, okay, maybe then in a few years, maybe we can record from the entire brain of the mouse. But there are reasons why this is maybe too optimistic or is not going to be happening. If you put that in relationship to where we are really, so this is the size maybe of the must recording. This is what we can achieve with the HIMS. And this is sort of the regions almost that you can give you access with the light beats. But as you can see, there is a lot more brain left to the image. And given that the difficulty of imaging the rest of it, especially the deeper layers, is going to be highly challenging, and they're even very difficult conceptually to come up with approaches, we started to think a little bit more on the high level, what is it that we need really? So what is it that really is needed, or how valuable would it be to have a single cell resolution whole cortical or whole brain calcium imaging in a mouse? I don't mean that it wouldn't be valuable, but quantitatively, how valuable would it be, say, compared to having access to half of the neurons or 20% of the neurons? What is the information that we gain or would be losing otherwise? So these are the kind of questions that I think are very interesting and we want to answer. And the reason for that is twofold. On one hand, I think it is important to be able to have a targeted way of focusing technology development on the things that are mattering most, because at the end of the day, what we want is to maximize the information that we extract from the brain. If there would be one metric, perhaps it would be maximization of bit per photon that we want to maximize. But also by asking this question, we might learn more about the architecture and the representation of information in general about the brain and the biology. So given an ultimate limit of resources, how should we allocate them wisely? So we started doing a little bit of that by just taking our data and doing some, for instance, coarse graining and looking at the information content. So I just want to give you one example. So we can maybe go back to this question that I was asking at the beginning. Now that I don't have maybe 100 neurons, what can we say about this? So we can ask how much information about the given neuron's activity is encoded in the population. And one way of asking that is to look at mutual information. So if we look, for instance, at the mutual information represented across these 5,000 neurons in the brain, we see that they exhibit a sublinear increase with the number of neurons added. But an interesting observation here is also that the amount of information very much depends on the state of the animal, whether the animal is in locomotion or is stationary. If we do now the same thing, let's say with the mouse cortical neurons, we see no saturation of mutual information interestingly, up to 2,500 neurons. And this observation holds up even to the much larger neuronal populations that I was showing you before. And there is an even a greater gap between the states of the animal that is performing, whether it's in locomotion, what kind of sensory inputs it's seeing, or whether it's in stationary constants. So I think these are the type of questions that we are interested in exploring further. We can now ask, if I then subsegment the volume, how is the information distributed within subsegments of the volume, and so on. And obviously I was drawing a very coarse picture with all of that, because the brain is obviously not a homogeneous collection of neurons. There are also a lot of different cell types, and the answers will also, amongst other, depend on the complexity and the dimensionality of the behavior that the animal is doing, which needs to be all taken into account. So with that, I hope I have given you a little bit of a flavor of the kinds of tools that we have developed and what kind of questions they could lend themselves to be investigated. And with that, I just want to thank everyone in the lab. I mentioned most people throughout the talk, that former lab members and current lab members that contributed to most of the project I mentioned. Just want to acknowledge our funding sources, and also want to let everyone know that we have various types of positions open currently in the lab. If you're interested in working with us, feel free to email me. Thank you for attention.
Thank you very much. Alipasha, are there questions within the audience? Otherwise, I'm just throwing the catch box around to Misha.
A really beautiful work, of course. So I have a question about, is this on?
Should be on, yeah.
Yeah. I have a question about sample movement. If you're imaging spots of 5 microns in size, so neurons are, well, in zebrafish, they're 5 microns, in mammals, they're a little bigger. So you don't need to image the information at 1 micron resolution, but the motion of the brain would benefit from seeing those fine edges and so on for registration and correction for intrinsic motion or sample drift. So how do you do that if you're using larger spots?
Yeah, so I think maybe I can elaborate a little bit more. So there are two aspects. So one is the size of the spot, and the other is the separation or the voxel spacing on how we position, how sparsely or densely we sample them. I think the latter is quite important to what you were saying, and the former is mainly important, I guess, if you want to say how can you maximize the amount of signal that you can generate. But we systematically investigate that, actually. For instance, in the last techniques, let me just see if I can have a slide that I can show you. That's obviously very important. So what we do is we start with dense and then go sparser, and then see where is the optimal trade-off. Okay, so maybe this is interesting. So here you can see, and what we look at as a metric is the F-score. So the way we deal with motions, there are two things. So one is the fact that you are fast helps a little bit with the motion. So the amount of motion you get is lower, but there is still some there. So we do non-rigid motion corrections and things like that. But also we investigate that systematically. So here you can see a spacing of 0.5 micron all the way to a spacing of 8 microns. And F-score is the probability of detecting neurons against the ground truth. And you see maybe up to something like 4 or 3 micron spacing, you're still in the same, you get a reasonable loss in F-score while still not being completely losing your information. And the other thing that helps is also taking advantage of the spatial, but also of the temporal information. So we take advantage of the time dynamics as well. So these things together computationally help us a little bit. Yeah. But it should be determined probably for each sample. For the zebrafish, I imagine it's different because neurons are much smaller, but also the volume is smaller. So probably one needs to be operating in a different regime. Thanks.
So Greg Fleischler on online has a question. Greg, can you
Should I go out of this?
Perfect. We can see you. You can unmute yourself and ask your question.
Okay, great. Alipasha, thanks for coming and thanks for a very exciting and interesting talk. I was curious at the end when you were talking about getting to a single technology for whole brain single cell resolution recording. And I wonder if while you're considering pursuing those types of technologies or thinking about them, you think it's also worth considering a course to fine approach that uses existing technologies that can image the whole brain, but at worse than cellular resolution, like starting with fMRI of the whole brain, identifying regions that are related to stimulus and behavior, and then using increasingly better spatial resolution technologies like light beam microscopy and the other techniques you mentioned to zero in on imaging only the regions that are most important to the behavior or stimulus at single cell resolution. Is it worth pursuing something like that in parallel to pursuing a single technology that does it all at once?
Yeah, absolutely. I didn't mean to argue by any means that we should only be pursuing one technology and whether that technology is light beats or anything else. I don't think with light beats you can get to those depths that requires for the entire imaging of the brain. So if you want to record from subcortical regions, most likely you need right now to use more invasive technologies if you want to look at single cell resolution. But using fMRI or even other technologies to identify the regions that are relevant for you is definitely something that is very useful and is also being done. The other is just wide field fluorescence imaging of the surface. So fMRI is something that we use with our collaborators who work in non-human primates in order, for instance, to identify the regions of the cortex that are sensitive to faces. And then we can go there with electrophysiology or with imaging and identify those. Absolutely, that's one way to go. I just want to point out also what I said at the end of the talk. Quantifying the amount of information that you are extracting is exactly has that in mind, because you can imagine even if you can have single cell resolution, there may be cases where you can trade off that if you can record from, say, a fixed number of voxels, you can still navigate the trade-off. Is it better if I keep single cell resolution but limit them to a smaller volume, or disperse them throughout the brain, or start with larger voxels and then cover a larger area? So those questions, I think, are best navigated by looking at the appropriate matrices that are need to be information-based that you're extracting.
Yeah, so we have more questions in the room. I'm just walking over.
Okay, it's really fantastic talk. I just have a question about the bees microscope. So previously, like you have like 1 to 4 kind of pulse going in, but now you need to squeeze 32 pulse into a window where you usually have 4. Do you need to do some specific things to manage the heat issue to avoid heat kind of loss?
Yeah, thanks for bringing up this question. That is a key was a key concern that obviously was on top of our mind. So I can say a couple of things. So one is that this excitation strategy with 1 pulse per pixel is relatively efficient, because if you consider that most people, when you use an 80 megahertz laser, are exciting the same voxel with multiple pulses, you can reduce the amount of average power per voxel by going through that. But definitely that still was a concern. But I think we were helped by a couple of factors. So one is that the fact that these foci were distributed across an axial range helped with the dissipation of the heat from the individual ones, and that's time scale. But most importantly, I think the fact that we are using an 8 millimeter window on top of the head of the animal helped a lot with the dissipation of the heat. So let me show you. So what we did were 2 things. So one is we did immunohistochemical experiments to look at the heat response of these neurons under various conditions. So for instance, when we have under 400 up to 450 milliwatts, when we distributed over a large area, it seemed to not induce any responses. Whereas we had the positive control conditions, where we had, say, 360 milliwatt, but within a much smaller field of view, we could induce the response of these neurons. So a lot of the thresholds that were previously established were based on small windows. So we investigated and see how far can we push things. And this was then also consistent with some modeling work. And I think Casper had done previously something related to this. So we see if you look at 400 micrometer field of view, you do get a much stronger heat response within the volume that you're exciting. But by going to these large excitations of 4 millimeter excitation within an 8 millimeter window, it really brings down the temperature. So there is a trade-offs that you can navigate here to be in this safe regime.
Are there more questions in the room? There are also no more questions online. I will just hand over the word right now to Robert Pribadil to give us a brief outlook of what's about to come next. So you can turn on your camera and maybe let us know which other talk we can expect next. Yeah, thank you.
Okay, yeah. Thanks, Ulrike. I hope everybody can hear me. And thanks, Alipasha, for really an excellent and impressive talk. So thanks on behalf of the EMBL participants, I guess. So in 2 weeks' time, it's EMBL's turn again. We'll be hosting this joint seminar series. And in 2 weeks, Carlo Bevilacqua, a PhD student from my lab, will actually present. And he will talk about our latest work in brilliant microscopy, and especially how we've recently optimized it to allow for 4-dimensional recordings of mechanical properties during embryo development. So if you're interested in this topic, join us in 2 weeks at the same time, as always.